Good afternoon, Siddharth sir.
Good afternoon. How are you doing, sir?
I'm fine. What about you, sir?
I'm doing well. Thank you.
Great. And thank you so much for accepting our invite.
It's my pleasure to talk to all of you.
What happened? Afternoon session was delayed a bit. Okay. So 5 minutes late. Sorry for that.
No problem. No problem.
Yeah. Shalaka, ma'am?
Yes, sir.
Yeah, I think we can start now.
Okay, sir.
Yeah.
Okay, sir.
Yes, sir.
Yeah. Good afternoon, dear all. I would like to take this opportunity to express our hearty welcome to our speaker for this session, Dr. Siddharth Jhunjhunwala, sir. Welcome, sir. Dr. Siddharth Jhunjhunwala is an assistant professor at the Center for Biosystem Science and Engineering, Indian Institute of Science, Bengaluru. He is currently a DBT Welcome India Alliance Intermediate Fellow and in the past has been awarded the Ramanujan Fellowship and R.I. Muzumdar Young Investigator position at IISc. In collaboration with the numerous colleagues, Siddharth has published over 40 research articles and has been awarded 2 international patents. His primary research interest is in the field of immunoengineering. Now I request Dr. Siddharth sir for his talk on engineering innate immune responses. Please, sir.
Thank you very much for the very kind introduction. It is my pleasure to be speaking to all of you and providing an overview of what we have been doing in my laboratory over the last 5 years or so. Thank you very much for the invite. So today what I'm going to share with you is mostly data that we have generated over the last 4 years, I would say, in my laboratory. The screen share is visible, right?
Yeah, it is visible,
yes.
Sir.
Fantastic. And is my audio okay? Can you hear me?
Yes, no problem, sir.
Fantastic. So let me get started, and I'll try to keep this brief. It will probably be a maximum 1 hour, but feel free to interrupt me if you have questions. I can't see your hands raised or anything, so if you feel like you want to interrupt me, please feel free to interrupt me. What we are going to do is provide you with an overview of 4 different projects that are going on in my laboratory and the progress we have made in it, with the general theme of trying to understand how innate immune responses function and how we might be able to modulate or engineer them. How does this relate to COVID-19 and how does it relate to this theme that you are being proposed, that you're coming for this refresher training for? Well, it relates in a few different ways. We don't necessarily work on COVID-19 directly, although there is a project that we have just recently started on some new vaccine strategies. But a lot of what we do, which is to understand and modulate innate immune responses, if we are able to kind of achieve that goal, we would be able to develop therapeutics for viral diseases like SARS-CoV-2. So it sort of relates back to that. And my first slide will try, in my first slide, I'll try to explain how this happens. So let me see if I can, yeah, laser pointer is a better option. So this is a recent review article that is a fantastic review article that came out on in Frontiers in Immunology that describes the interactions between COVID-19, the disease itself, and neutrophils, which are one of the major players in this disease. So just to take you through, this is a very busy slide, but let me take you through the top part first. So what we understand about neutrophils is that they are generated in the bone marrow from a granulocyte monocyte progenitor cell and eventually form these neutrophils, and they're identified using various surface markers. These are not as essential for you to remember as of now. We'll keep coming back to them in my presentation, right? And in the blood, these neutrophils sort of from the bone marrow are generated, and then they are removed, come out into the blood, and sort of homing into the blood, where they are present at the homeostatic conditions. These homeostatic neutrophils under inflammatory stimuli, for example, an infection in the lung and bronchiovascular space, they go into those sites and then take part in certain reactions, but they do not have any specific receptors that would recognize pathogens apart from the TLRs. So they may sometimes non-specifically damage a large number of different tissues as well as cells that are infected, right? And as cells age, so from the homeostatic neutrophil that can get out into the tissue space, these cells can age eventually, and the aged neutrophils get back into the bone marrow for death and basically apoptosis and uptake by other immune cells. But in severe COVID-19, this whole process is hampered. And we'll show you that this is not just in severe COVID-19. In fact, we have been talking about this for the last 4-5 years, even before the pandemic started, that neutrophil responses can be hampered in a variety of inflammatory conditions. What ends up happening is the body does not wait for the mature neutrophils. See here, the whole maturation cycle that's happening, and then only the mature neutrophils are coming into the blood. Whereas here, because there's an acute need for neutrophils in the blood, the immature neutrophils at every stage are coming out into circulation. These immature neutrophils as well as mature neutrophils now respond to infections at the bronchoalveolar space in the context of COVID and cause destruction of the virus itself, the cells that carry the virus, but also the neighboring tissue, thereby causing major damage in the lung. And there are autoantibodies then that are generated in response to the damaged cells that are present in the lung. And there's a whole cascade of inflammatory reactions that occurs that we cannot deal with, and the body sort of succumbs to it, right? So everything sort of begins at the neutrophil stage, and we try to prevent all of that using various sort of therapies. You would have seen corticosteroid usage that prevents neutrophil extracellular trap formation. You've also seen some activated platelet therapy that prevents neutrophils, activated neutrophils from responding. And then you also promote neutrophils to become aged and move away quickly into the bone marrow. And there are various sort of drugs that can be used for that. Now, this is the context in which I'm going to describe all of my projects, except we're not going to be talking about COVID-19, but we're going to be talking about drug delivery systems, about biomaterials, and how some of them can replicate some of the things that are happening in severe COVID-19. Okay, so with that, let me go to the first project that we are working on in my laboratory, which is how do we take neutrophils to their limit, which is what happens in COVID, right? So how do we do that experimentally in an animal model system so we can understand the neutrophil response better and develop new therapeutic strategies, new treatment strategies for them, right? So that's what I'm going to explain. So as shown in the previous figure, progenitors, which are basically stem cells or hematopoietic stem cells or neutrophil progenitors, are producing neutrophils that get out into circulation normally, and then they eventually go into the tissue upon activation, right? So they get activated and they go to the tissue site, and eventually they get utilized there, or if they're not utilized, they come back into the bone marrow and they are degraded. Now, under activating conditions, when there is an inflammatory stimulus, the progenitors can expand because there's more neutrophils required at the tissue site that send signals to the blood. So more neutrophils are present in the blood, which signals to the progenitors that more neutrophils need to be produced, so they divide faster. This is normally what happens in a single stimulus or a small number of stimuli or a very low grade inflammation, which may happen where a very weak bacterial infection or even a simple viral infection that causes your common cold, right? This is what happens. But what happens in a severe infection or where there are multiple stimuli, basically the amount of inflammatory stress, either because of the viral load or the bacterial load is because it's very high, what happens? This is the question that we want to ask in my laboratory, and this has not been answered. And so here I kind of show you what could be happening. If you look at neutrophils numbers in terms of total numbers per activity, per number of stimuli, and the x-axis being the number of stimuli and the neutrophil numbers or activity in the y-axis, will they continue to increase forever? So that is, will there be more and more neutrophils here, more and more neutrophils here, or will you see a plateauing and then there's a loss of activity? What really happens? That's the question we wanted to answer, and this has implications for diseases like COVID, right? To answer, what we did was we decided to use a biomaterial system. These are scaffolds with biomaterials that cause the local inflammation. Why did we use this? Well, if you induce infection, there are a lot of other immune cells that get involved in the whole cascade of inflammatory reactions. There are B cells, T cells, there are, of course, neutrophils and dendritic cells and monocytes that start the whole reaction, but there are adaptive immune cells, NK cells that get involved with infectious agents. And that causes various complications for us to really understand what neutrophils alone are doing. In the context of inflammation, if you put in a biomaterial implant, you have high-grade inflammation. You can control the grade of the inflammation by putting different types of implants. And the primary responders to these materials, especially their synthetic materials, are neutrophils and monocytes. There's very little reaction from B cells, T cells, and NK cells. So it's an easier model to kind of use to study what happens. And we could do this in humans and animals. I'm not going to talk to you about the human work today because that's still ongoing. Hopefully, in about 2-3 years, we'll have much more data in the human space for us to be able to tell you what's happening in that front. But in the animal space, we do some studies with experimental mice. And in those mice, we implant microspheres. These are just a model for generating inflammation. But these microspheres have various functions. They could have various functions. They can be used for drug delivery. They can be used for tissue engineering. They can be used for improving implant activity for strength, bone implants, for cartilage implants, and so on. We use 2 different materials, alginate and chitosan, and I'll explain to you why we use 2 different materials in the next slide. Basically, alginate is somewhat biocompatible. When I say that, I mean every material is somewhat biocompatible. Many of them are to some grade are compatible. Well, what I mean by this is that alginate has a low-grade inflammatory response. There are fewer immune cells that respond to alginate. The total number of cells that are being produced in the body are also fewer. Compared to that, the chitosan has a very high-grade inflammatory response. Shown here is a H&E section because you can't really see individual spheres like alginate. And these H&E sections are basically microspheres in pink embedded into tissue where there's a large infiltration of neutrophils and monocytes that surround these chitosan particles, right? So this is the low-grade inflammation. This is the high-grade inflammation. And this now can be used to model and understand how neutrophils behave at various sites. So we'll be using this marker Ly6G in mouse to identify neutrophils. And this is just an understanding of how neutrophil numbers change in the bone marrow, blood, and the peritoneal space. This is where we implant the microspheres, right? This is just a place in the body where we can easily implant microspheres and understand the whole inflammatory cascade. And as shown here, if you look at mock surgery, which is just surgery with and then no other material implanted, just saline being put in, or if you put in chitosan microspheres, or if you put in alginate microspheres, there are no major changes in the total number of immune cells or no total number of neutrophils in the bone marrow. Again, in the blood too, there's no major change in the total numbers. The total numbers of neutrophils are very tightly regulated at these 2 sites. But at the inflammatory site where the implant is put, right, this is a tissue, the numbers are not tightly regulated. So normally there are very, very few cells in the mock implants, and this is percentage. We can convert this to numbers too, is the same idea. But you will see a large percentage of neutrophils in this high-grade inflammatory response in chitosan, and a relatively high number of neutrophils in the low-grade inflammatory response in alginate. Right? This sets the setting for what's happening in terms of after the biomaterial is implanted, what happens to neutrophil numbers. Right? So basically, we're saying there are little to no changes in percentages or numbers in the bone marrow and circulation, but at the tissue site, you see a large number of cells. Right? So now let's look at what's happening in the bone marrow. So you don't see a change in total numbers, but is this replicating? Is this a good model for what happens in an infection setting where the bone marrow starts proliferating, the progenitor starts proliferating to produce more neutrophils? For this, we use advanced flow cytometry, identify HSPC, which is hematopoietic stem and progenitor cells, and also GMPs, which are granulocytic monocytic progenitors, which are neutrophil-specific progenitors, and then calculate the numbers of these. So the percentage of GMPs, which can again be converted to numbers, in the bone marrow, in the chitosan implants in orange, there's about a 10-fold increase at day 10, whereas in the alginate, there's no major increase in the numbers of these GMPs. In the spleen, similarly, there's about a 100-fold increase at both day 7 and day 10 in the chitosan implants, but not as much in the alginate in comparison to the control, which is just a mock or saline surgery. What this goes to establish is that we do not have major changes of neutrophil numbers, but their phenotype, which I've not shown you here, but they are changing, and the numbers of progenitors are changing. And this is termed as emergency granular poiesis in the literature. Excuse me. So we basically have a large number of new neutrophils that are being produced. The number of neutrophils in the bone marrow and blood are tightly regulated, so they're not changing much, but the numbers in the tissue are increasing dramatically. So what exactly do we do next? Right? So we decided to track neutrophils now, spatially and temporally. Spatially is in different locations in the body, temporally being across over time. what happens to neutrophils? And does this model resemble what might happen in infection cases? And for the spatial and temporal tracking, we utilize this particular model. This is again in mice, in animal models, where we surgically implant our biomaterials, either the chitosan or alginate implants at day 0. And we allow 5 days of rest time, where the surgery itself, the wound that the surgery causes is fully healed. So there are no responses because of surgery. Whatever responses that are there are because of the biomaterials. At day 5, we inject this molecule known as EDU, which is basically a uridine analog, and it is easy to label cells that are dividing with this EDU. So all newly dividing cells take up EDU, and we can then get in an antibody or a small molecule that will bind to EDU, and then we can identify all the cells that are newly divided. So there's a way to understand what are the new cells that are being produced, and then we euthanize animals at different time points, retrieve cells from the bone marrow, blood, and the peritoneal cavity. We also do it from the spleen, but I'm not going to show you that data. The peritoneal cavity is where the implants have been placed, and identify how many of these cells are newly dividing cells, right? And this is sort of the flow cytometry plot. We identify all the live cells. Among the live cells, we look at only the neutrophils, which are Ly6P positive. Among the neutrophils, we look at those that are EDU Cy5 positive, which basically means that are newly dividing cells. Some of the other neutrophils will be the old cells that were already present. They're not newly present coming in, right? And this is a large amount of data in one slide. This is what happens in the bone marrow, blood, at the inflammatory site, in saline implants, in chitosan implants, and alginate implants at up to 7 days post injection of EDU, right? Now let's look at this data one at a time. The most important data points are the day 1. You can already see at day 1, and this is statistically significant, in the bone marrow and in the blood, the orange dots, which are the chitosan animals, have higher numbers or percentages here of labeled neutrophils. That is, there are more numbers of these labeled neutrophils showing up much earlier, day 1. At day 2 and 3, the same chitosan mice have higher numbers of labeled neutrophils showing up in the blood and the inflammatory site much earlier. Day 2 and 3 is showing up, whereas in the green and black, which is the alginate and saline, they're showing up closer to 4 days here and 4 days here, right? Suggesting that there's a change in the kinetics of neutrophil recruitment, right? We use a mathematical model, which is similar to some of the pharmaceutical models that you may have developed in your classes, to find out what percentage of cells are labeled and what this means. I won't get into the details of the model itself. It's a basic, simple differential equation-based modeling, and that gives us this sort of data. When we fit the model to our data, we get these 3 important data points. This is the most important data points. Maturation time in the bone marrow. Remember, I showed you in the first slide that the maturation time reduces. There are more immature cells coming out into the circulation in COVID-19 infections. Similarly, in our chitosan implants, the maturation time in the bone marrow drops from normally about 50 to 55 hours, which is what is normal in saline implanted mice or non-implanted mice. Compared to that, it's half the time frame, meaning many immature cells are coming into the circulation in the chitosan implanted mice. In comparison, the low-grade inflammation, which is caused by alginate, doesn't really lower the maturation time by much. There's some change, but it's not significant, but it's not dramatically different, right? Correspondingly, you will see changes in half-life of the cells in the blood. This is cell half-life, neutrophil half-life. Normal half-life is known to be about 5 to 6 hours. We are showing that in our saline implanted mice, so at 4.7 hours, within the normal range. Whereas in the chitosan implants, it drops about twofold to about 2.8 hours. In the alginate, there's a slight increase, and we're still trying to understand what the slight increase is, but this is well within the range of error, and the saline and alginate are very similar. And at the inflammatory site, which is where the action is taking place, normally we'll see very short half-lives in the saline, and even shorter half-lives in the low-grade inflammation. But there's a doubling of the half-life in the chitosan implants, suggesting that the neutrophils are playing a much larger role at the high-grade inflammatory site. So I'll just read out the implication of this, basically this data, that emergency granulopoiesis, which is occurring in the chitosan implanted mice, causes a twofold reduction in bone marrow maturation time of neutrophils, right, this time frame, which suggests that there are immature neutrophils in circulation and in the bone marrow. And these neutrophils spend less time in circulation, but more time at the inflammatory site here. And this is sort of clear. This suggests that There'll be immature cells in circulation, which may not be as reactive, but at the inflammatory site, they spend more time, so they may be doing more damage when that happens, right? To confirm, we actually looked for immature cells, which you can do using this marker called CD101. And you will very clearly see here that this is showing mature cells. So this is 100 minus this is what we should consider as immature cells. So you see much lower numbers of immature cells in the chitosan and bone marrow, in the blood, and at the inflammatory site. Inversely, this means much higher number of immature cells are present at all three sites in the presence of chitosan implants, suggesting that they are going to respond very differently. Right? So a quick summary of this project one, we basically see that material-specific changes are present in global neutrophil responses following biomaterial implantation. And this model now can be applied to understand what neutrophils might be doing under inflammatory conditions like bacterial infections also. The neutrophil kinetics that we have measured, this is basically a two-fold drop in maturation and circulation time and inflammatory site residence time. That's what I talked to you about before. This also is a very important thing for biomaterials and drug delivery systems because the compatibility, which is normally only measured at the inflammatory site, may need a rethink. Basically, a biomaterial that's implanted at some tissue site is causing a change in the bone marrow. So that's a big, big change, right? So we may need to rethink what compatibility actually means. All of this very talented graduate student in my laboratory, Alkesh, who's shown here, with help from Thiruvikraman, who's another graduate student who did all the mathematical modeling. And this work we have put up in bioRxiv, and it's actually the work just got reviewed and it's close to publication. It should hopefully come out in the next month or two. Right? So that's our first sort of setup of what happens to neutrophils when we really take them to their limit when we implant large materials. Now, if you think of biomaterials in drug delivery systems, there are various scales, right? These are the large materials, the macro scale materials used for tissue engineering and some of them for bone regeneration and so on. But there are also micro to nanoscale materials. What happens to them? They are normally phagocytosed by macrophages and neutrophils. So we wanted to look at what happens if you take these to the limit too. So basically, this is the question that we were interested in asking. Let's take a phagocytic immune cell, whether it's a macrophage or neutrophil, doesn't matter, and give it a large number of particles. Now, this cell is saturated with a large number of particles, and then they ask the question, what happens to the cell? What happens to the cell as in functionally does the cell change in its activity? And if it does, then that could have implications in, again, bacterial infections and viral infections, something that I'll show you some data for. All right. So how do we ask this question? Again, we used a model system because, again, using bacteria causes lots of different activation of different immune cells. So we used a polystyrene micro and nano particle for some of these studies. And this is the type of study that we did, which is a sequential phagocytosis experiment, where we take phagocytic cells in culture. We provide one set of fluorescently tagged particles, shown here in orange. So there'll be some cells that will take up particles, some cells that won't. Some cells will take up a large number of particles, some cells won't take up any. Now we provide another set of polystyrene particles with a different fluorophore. Now let's call them green in color, right? So they're different, we can easily identify them. If you take both of the, now the question that we want, if you take both these cells, this is what can happen. The bystander cells, which didn't take up any of the orange particles, they could take up some green particles, or the cells that took up the orange particles could take up green particles. This idea is called sequential uptake. Sequential uptake being the cells that took up the first set of particles taking up the second set of particles. Bystander uptake is cells that did not take up any particles taking up the new particles at the second round. Right? So we really compare bystander to sequential uptake and understand how functional are these cells after they have taken up particles. And here's the first set of data. When you take 500 nanometer size polystyrene particles and check across various cell lines, raw 264.7 is a mouse macrophage cell line. HL60 is a human neutrophil-like cell line. We also tested with human monocytes isolated from venous blood and human neutrophils isolated from venous blood. In all four of these cases, and in fact, this is actually now statistically significant, we repeated it enough number of times now. The sequential uptake, shown by this short form SEQ, is much higher, significantly higher compared to bystander uptake, right? That is, the cells that have taken up one set of particles first are much better at taking up the second set of particles compared to cells that never took up the first set of particles. This is not true only for one size of particles. It's true for all sizes of polystyrene particles and across modifications. Whether you have COH on the surface, you have no modification. These are all non-modified particles. Whether you have PEG on the surface, and now we have data for silica particles, we have data for iron oxide. In fact, we've tried many materials, and this holds true across all material types. And this is not just an in vitro phenomena, it happens in vivo too. We check this with Drosophila hemocytes, which are phagocytic immune cells similar to the macrophages. There too, we see sequential uptake being higher than bystander uptake. We also see this in an animal model in mouse, in vitro and in vivo. Both the neutrophils and the macrophages take up larger number of particles when they've already taken up one set of particles compared to the bystander cells, which have not taken up the first set of particles. Right? So what does this mean? Why is this happening? What could be what does this all imply? In bacterial systems, it's well known that once you activate an immune cell, when you activate neutrophils or monocytes, they can take up many more particles because the cells are activated. So are we really activating cells here? Right? And our data, basically, I'm showing you the result, stating the conclusion, but let me show you the result. It's not a result of cell activation. To prevent cellular activation, we block LPS-mediated stimulation. So there's no stimulation here. We block it using this drug called TAC242. Here is a positive control of this is the positive control of LPS alone, and this is a positive control of LPS plus TAC242, so blocking the positive control. In all four of these, and this is all statistically significant, we see sequential uptake being higher than bystander uptake. Basically, whether you activate the cell or you do not activate the cell or you suppress activation, in all conditions, you are going to see that sequential uptake is significantly higher compared to bystander uptake. We confirm this also by measuring cytokines that cells normally produce upon activation. So if you provide LPS, the cells start to produce a large amount of IL-1 beta, TNF alpha, and IL-6, very clearly seen here. But when you just add particles and compare it to naive cells that have not seen particles, you don't have any large amount of cytokine being secreted. This goes on to show that our particles are not activating cells, but still somehow those cells are much better at taking up the next round of particles or the next round of bacteria and so on. We'll come to that data, right? So the other question that some of our colleagues asked, an interesting question: what if there is heterogeneity in cell population itself? It's well known many immune cells are heterogeneous. Even the neutrophils, some can be more phagocytic, some can be less phagocytic. So you're just seeing that change here, right? So to overcome that phenomena, what we did was we first, it's a slightly complicated experiment, so let me take you through it. We took these cells in culture. We fed them with one set of particles, let's say pink particles now, right? So you'll have some cells that have taken up pink particles. If there was heterogeneity, those are the phagocytic cells, and the other cells that did not take up pink particles, they were particle negative, and we can sort them. Use fluorescence activated cell sorting to separate the particle negative and particle positive cells. Then we take them through a sequential phagocytosis experiment, which is give them orange particles and then green particles, and measure how many take up orange, how many take up both orange and green, and how many take up only green for both the particle positive and the particle negative cells. And you'll observe this data. This is for the particle positive cells, and this is for the particle negative cells. Yes, is there a question? Okay, so I guess there's no question. You can hear me still, right? Hello?
Yes.
Oh, okay, thank you. Yeah. So in the particle positive cells, you would expect sequential phagocytosis to occur. That is, those cells are already phagocytic. But in the particle negative cells, if there's heterogeneity, those cells are not phagocytic. But here we're showing actually they can be phagocytic, and in fact, sequential phagocytosis is still grown to be greater than bystander phagocytosis, right? So why exactly is this occurring, right? So that's when we came up with this hypothesis based on some literature that exists, that phagocytosis could actually induce changes in the cell membrane. And that's what we actually are seeing. There are changes in cell membrane fluidity. So these are cells by themselves. Their fluidity that we are measuring using this Laudan dye, don't worry about the assay itself, but it measures fluidity. The higher this value here, our membrane is less fluid. And the more fluid it becomes is here, which if you treat with a cholesterol removing agent called M-beta CD, you will see the cells becoming more fluid. So this is a positive control. These are negative control. And as soon as you give them particles, 500 nanometer particles at 1:50 ratio, 1:200 ratio, you will see that the membrane has become more fluid than the controls, but not as fluid as the positive control. So it's fluid, but not extremely fluid. And this also is measured by cellular stiffness. So with particles, our membrane's cellular membrane stiffness is lower compared to cells without particles, cells by themselves, that is. So all of this suggests that phagocytosis does not necessarily need to activate cells, but can lead to changes in the membrane, which result in sequential phagocytosis. So, okay, I've been talking about sequential phagocytosis for a long time. How does all this matter? Well, it matters because now these cells could be different at clearing bacteria. Here it is bacteria, it could be applied to viruses too. So we did an in vitro experiment where we took cells by themselves or cells with particles and then added E. coli to them and measured how quickly is E. coli removed. So at 2 hours, we added E. coli for 2 hours. So we see that the cells with particles take up more E. coli, which is expected, right, because of sequential phagocytosis, compared to cells by themselves that take up E. coli, but they don't take up as much. Then we removed all the E. coli and followed the numbers of E. coli. And you will see at 6 hours, the numbers of E. coli in the cells with particles continue to remain high. But as you come down to 18 hours, the numbers of E. coli in cells with particles almost equivalent, in fact, slightly lower than the cells by themselves with E. coli, right? Which, if you convert to the percentage bacteria remaining, is statistically significant lower percentage of bacteria remaining. More bacteria are present here at 2 hours and very few were left at 18 hours, suggesting better killing capacity. We show this also in vivo in a mouse model where we inject particles followed by bacteria in the peritoneal space and look at bacterial numbers in the peritoneal fluid, in the peritoneal cavity cells, spleen, and in the kidney. In all 4 sites, we see lower numbers of bacteria at 2 hours and 6 hours to some extent, and statistically significant in certain instances. In this mice in which both particles and bacteria were injected compared to only bacteria, suggesting that the sequential phagocytosis phenomena can be used to improve bacterial clearance capacity of immune cells. This is not true only for E. coli, which is a pathogen that's removed by macrophages, but also for Staphylococcus epidermidis. This is not epidermis, it's actually epidermidis. Sorry about the spelling error. Which is primarily removed by neutrophils. The same idea injected in the peritoneal cavity. And in fact, you don't see any bacteria going to the kidney when your particles are injected first. That's a cool thing. There's no spread of bacteria. We also see less bacteria in the spleen. We see less bacteria in the peritoneal cavity and in the peritoneal fluid, all suggesting that using particles now we can quickly remove bacteria from in vivo settings, thereby promoting faster clearance, basically. So this work was done by a very talented student of mine, Preeti, who's actually graduating now. She just submitted her thesis. Basically, her work shows that phagocytosis drives additional uptake in phagocytic immune cells, and this can we've already shown some data that it could be utilized for enhanced clearance of bacteria. We plan to test this also for enhanced clearance of apoptotic cells that could help in wound healing applications and in anti-tumor activity. All of this work is also recently actually been reviewed. We got very positive reviews from a very good journal. We're resubmitting the paper soon. You can also see all of our data on this bioarchive where we have posted the paper. So that's with the second part of the talk. We basically taken you through what happens when we have taking neutrophils to the limit or innate immune cells to the limit, using either macro scale material like an alginate or chitosan implant, or a nanoscale material like a polystyrene particle. In all of these cases, we show that we can modulate neutrophil and macrophage activity, and this may have implications for diseases, inflammatory diseases, especially infectious disease-related inflammatory diseases. Now let's take a different turn and look at these innate immune cells in a more in a more specific context of disease. And the disease that we were interested in was diabetes. Again, diabetes has relevance to COVID because many diabetic patients are the ones who suffer from severe COVID. Why is that the case? This is something that we were interested in from before from a diabetic foot ulcer perspective, right? So we wanted to understand how to alter neutrophils in diabetic foot ulcers. It's known that neutrophils in diabetes have reduced chemotaxis, phagocytic activity, their microcidal activity is reduced, and their ability to use ROS is also altered. When we started this research, just when we were starting it in 2015 to 2016, there were these 2 seminal papers published in wound healing of diabetic foot ulcers, which showed that diabetes primes neutrophils to undergo this activity called netosis, which delays wound healing, which impairs wound healing. So the question we were trying to ask is, can we understand what happens to neutrophils in diabetes that causes them to become more reactogenic or more activated in diabetic foot ulcers? And then now can we target neutrophils to reduce or improve healing capacity in the individuals with diabetic foot ulcers. So this is a clinical study that we did in collaboration with Karnataka Institute of Endocrinology Research, which is a tertiary care government hospital center for diabetes and diabetic foot ulcers. Here is a patient that we recruited into the study, and we are using a camera to figure out the wound area, which is important for us to understand whether wound healing is occurring or not, right? And if wound healing doesn't occur, unfortunately, for many of these individuals, there'll be amputation, and that leads to morbidity and mortality, right? So this is the actual healing rates that we measured in that clinic, which is similar to the healing rates worldwide. It's been reported by others too. This is stage 1 or grade 1 ulcer, grade 2 ulcer, and only about 66% of grade 1 ulcers heal with current state-of-the-art healing strategies, and about 62% of the ulcers with grade 2 heal in the current state-of-the-art healing strategies, right? So the question we were asking is, why do these ulcers, other ulcers, in 33 to 38% of the individuals not heal? So we recruited individuals who had diabetics that were diabetic with foot ulcers. We also recruited individuals with diabetes without ulcers and had healthy controls. And for the first one, we took biopsy sample of the ulcer itself. But for all three, we took blood sample and did biochemical and clinical tests to understand what is happening. And I'll take you through all the data. So this is just the clinical characteristics of those with foot ulcers, right? These are stage of ulcer 1 or grade 1 or grade 2. And the age, BMI, HbA1c, FBS, PPBS, all are very similar. So there's no differences between the two grades. They're just, the surgeon grades these ulcers. So we're just showing that in this context, none of these clinical characteristics are different. This is the lower confidence interval and upper confidence interval. This is the median. So you can just quickly compare the median and you'll see there's not much of a change, right? But they're highly diabetic, as you can see with the HbA1c values. Now we start different data. So this is all our data. This is all the patients that we recruited with foot ulcers, right? Some of them healed. So those are shown here in blue. This is a heat map. It's a very different representation of the data. And these are the people who these ulcers did not heal, right? And we have all this clinical data. Then we have some biochemical data. And we have a large amount of immunological data on neutrophils, markers among neutrophils, markers among monocytes, the CD14 positive cells. And basically, if you do a univariate analysis, which is a simple, let's take one marker and see if that marker is sufficient to explain healing or non-healing, the data suggests that no one marker is good enough. But some immunological markers are starting to become good enough, right? So we did what is known as a high throughput analysis of we did about 20 color flow cytometry to understand all the different markers in this, showing how immune cells in healthy individuals look. In diabetic individuals, you see that the immune cells look different from the healthy individuals. In individuals with stage 1 foot ulcers, they are showing different than the diabetics. And in stage 2, again, it's very different from the diabetics and the healthy and the stage 1. So this suggests that the immune cells are altered in these diabetic individuals, those with foot ulcers, right? And 3 different markers on monocytes were okay predictors. I say okay because they were statistically significant, but not great in terms of all the other details in terms of statistical markers. This is what is known as a receiver operator characteristic or receiver operator curve. And the area under the curve tells us how good the measurement is. We usually want area under the curve to be above 0.75, above 0.8 to be considered a good predictor. But CD63 on monocytes is not a bad predictor individually. HLA-DR again on monocytes also individually is not a bad predictor. It's not a great predictor, but not a bad predictor. Same thing with CD11B. So this all suggested that there are immune cells that are differently activated in individuals with diabetic foot ulcers. Now, what we did was we tried to use a very different statistical learning approach. This is similar to machine learning, but machine learning is completely agnostic to the data. Here we use logistic regression. And then we did this receiver operating curve, and we noticed that a combination, what is known as a multivariable logistic regression, if we do, we have a much better prediction, as you see here with AUC of 0.87, in predicting how some of the ulcers are not healing. And so basically what we found is that there are 4 markers in combination that are very good. It's only in combination individual. In combination, they're very good at predicting non-healing. That is, total cholesterol, LDL cholesterol together, along with the CD63 expression among monocytes are good predictors. To take LDL cholesterol, what this suggests, our data suggests, the higher the LDL cholesterol, which is the odds ratio, the higher the LDL cholesterol. There's a one step higher in LDL cholesterol results in a 18 step higher in non-healing. In comparison, the lower the CD63, the worse the outcome of non-healing. That is, one step lower in CD63 means a 88% chance of worse outcome in healing. So together, these markers tell us that what could be a good biomarker for the clinician to determine which ulcers will not heal, which is a very important point. Right now, if you go to the clinic with a diabetic foot ulcer, the surgeon and the endocrinologist cannot tell which ulcers will heal and which will not. They try all the treatment strategies, but some of them are not going to heal. So what are data? Preliminary data in 52 individuals suggests that a combination of these 4 markers can be used to predict which ulcers will not heal. If you use this now, the clinicians can provide more aggressive treatment strategies to them, better counseling to them, to tell them that they need to take better care of their foot ulcers. Right? So this is the human data. We started looking at seeing if we can provide now a better treatment strategy. For this, we moved to a mouse model of diabetes. And we see some similar changes. It's not the exact same because the mouse and human is very different, the systems itself. But we see similar changes in a diabetic mouse model. It's a leptin receptor knockout. So these mice are obese and diabetic, which is similar to type 2 diabetes in humans. Right? We look at neutrophils and monocytes. Neutrophils are positive. Monocytes are Ly6E high. Monocyte numbers are higher in these KO knockout animals, which are diabetic animals, compared to percentages and counts are higher in circulation. Same thing with neutrophils. Percentages and counts are higher in circulation compared to the wild type animals, which are the non-diabetic, non-obese. Right? Further, we can show, this is back to the first project, right? Immature neutrophils are higher in these diabetic animals. In the KO animals, the immature neutrophils shown in black bars are about 10% of the total neutrophils, whereas normally they're only 2 to 3% in the wild type animals. So they're significantly higher in the diabetic animals, again suggesting there's differences in neutrophil activity. And the activity can also be measured using their ability to produce enzymes called MPO myeloperoxidase, neutrophil elastase, and then all of these are also different in the knockout animals compared to wild type animals. And for the want of time, I'm not going to go into every piece of data, just going to tell you that there are differences in activity. So now that we know that there are differences in activity in the neutrophils and they are more immature, they're probably spending more time at the tissue site, at the wound site, we decided to now see if you can modulate the immune response, reduce neutrophil and monocyte activity locally at the wound site. And for these, we developed a drug delivery bandage using chitosan, because chitosan is normally used for diabetic wound healing in even humans. Non-cross-link scaffold looks like the non-cross-link scaffold does not absorb enough wound fluid, enough water, so it's not very commonly used in the clinic. We developed a TPP, a tripolyphosphate cross-link scaffold, which works much better in the mouse model at least. And we were able to show drug release profiles of vapomycin and tetracycline. Vapomycin is a well-known immunomodulatory agent. Tetracycline is an antibiotic primarily, is also known to be an anti-inflammatory in certain instances. So we wanted to test both of these, and we saw that both of these in the chitosan bandages release slowly. The tetracycline comes out rapidly actually within 2 days, which is still a good time, right? 2 days is still a slow release. Whereas the rapamycin can come out over 5 days, which is more than sufficient because these bandages are changed every 2 to 5 days. So this is very well within the limit of what we would like to use, right? So we tested this in the diabetic model where we created skin wounds. This is what the mouse model looks like. In the wild type animal, if you create a skin wound and put a splint that prevents contraction-based healing, you will see that most of the wounds heal by day 10 to day 13. This is normal. Compared to that, if you take the diabetic mouse, most of the wounds take at least 3 weeks to heal, about 1 week extra time to heal. Whereas if you put low dose rapamycin versus high dose, low dose heals within 13 days, very similar to wild type, right? We can quantify this data too, and this is quantified data from about at least six animals in each group, and that's why you see the large error bars. Look at the statistical significance. The low rapamycin treated groups are better than the high rapamycin treated groups. They are better than the blank, and they are actually not very different from the wild type. So wild type to rapamycin low comparison is not significantly different, suggesting that the rapamycin low, shown here in green, and this is seen here even in the data, is very similar to wild type healing at least by day 13, whereas the other two are not as good. When you compare it tetracycline 2, tetracycline was also done at low and high concentrations. It shows medium healing compared to the blank, but rapamycin low is still better than tetracycline and blank. So all of this suggests that immunomodulatory strategies could be used to promote wound healing in cancers. This work was done by a student of mine, Jayashree, who's also graduating soon. She's just done her colloquium and is submitting her thesis in the next few weeks. Basically, what she shows is that treatment of diabetic foot ulcers in the clinic is very different. There's human data, clinical data. The combination of these 4 factors could be useful in predicting non-healing for the clinicians. And we have developed an immunomodulatory strategy to treat diabetic foot ulcers. And this is in now 2 papers, both under review at different journals. Right? So let me take another 10 minutes to just talk to you about one final area of work in my laboratory, which sort of follows this diabetes work. It's not diabetes, but it's a very different model in brain tumor. The same idea of using flow cytometry to identify how innate immune cells, neutrophils and monocytes are very different in the tissue compared to how they are in circulation. So just to give you some background, we're looking at, this is all human samples, brain tumor samples. Glioblastoma is a very aggressive type of brain tumor with very poor prognosis. Median survival time is less than 15 months being 1 year and 3 months. And by 5 years, less than 1% of the patients diagnosed with glioblastoma survive. So it's really, really very debilitating and a rapidly spreading disease. Right, and current treatment strategies just do not work effectively. There is surgery, there is radiation therapy, there is targeted therapy, but targeted therapy, immunotherapy, does not work very well. That is because there's a lack of understanding of myeloid cells in the tumor microenvironment of the glioblastoma. Compared to that, in ovarian carcinoma, breast cancer, or lung cancer, we have a much better understanding of the immune cells in the microenvironment. So we set out to really understand immune cells in the brain tumor microenvironment and compare it to what is there in circulation. For this, again, we use this characterization technique of flow cytometry. We identified all the immune cells in the tumor. We also identified specifically neutrophils, which are CD15 high. These are other granulocytes like eosinophils and basophils. And then we identified 2 different monocyte populations. One's a traditional monocyte population, another new monocyte population that expresses CD15 to some extent. In GBM, that is grade 4, and compared to grade 3, and here you don't see a major difference in the tumor. But in the blood, compared to healthy controls, in the in the tumor that in healthy no tumor patients, we don't have any tumor sample. This shown in black. But in the circulation, we have blood samples taken from all these individuals, and you will start to see neutrophil numbers are much higher in individuals with these tumors compared to non-tumors, right? And in comparison, the monocyte numbers are actually slightly lower. What does this imply? What does this mean? So we did a thorough 3-dimensional analysis of immune cells across time and space in the tumor tissue as well as in the blood, right? And this is sort of a heat map. A lot of data, don't need to go through all the pieces of data, but just kind of showing that there are certain markers that kind of correlate with types of tumor. So the GBM individuals, and shown here, you can't really read, but there are these markers, CD36, CD66B, CD16. All of these are different in the grade 4 tumor compared to grade 3 tumor. But the most important of these markers was possibly the CD163 marker. CD163 positive monocytes are known to be M0 or M2 monocytes that are involved in immunosuppression. And we showed that the percentage of CD163 positive traditional monocytes and the new monocytes that we identified was much higher in the tumor tissue, suggesting some sort of suppression in grade 4 tumors compared to grade 3 tumors. Right? These are more aggressive, these are slightly less aggressive. So grade 4 tumors have much higher levels of these suppressive monocytes. We confirmed this with tissue histology too from the tumor tissue, and this is the quantification of the tumor histology. GBM patients, grade 4 patients basically, have much higher levels of these CD163 expression compared to patients. Interestingly, this CD163 expression is not very different in monocytes from blood or the neutrophils from blood, suggesting that there's some microenvironmental changes in the tumor that is causing these monocytes that are coming from blood to switch to M0 or M2 CD163 expressing monocyte in the tumor tissue, which is important because that can now be targeted maybe in the tumor tissue as a treatment strategy. We also noticed that only a certain number of correlated from blood to tumor. So monocytes and neutrophils, this is comparison. This is what is known as a correlation in blood and tumor. Only CD86 expression and CD63 expression to a large extent correlated from blood to tumor. Neutrophils and monocytes in blood were resembling those of tumor. And it's well known that high CD86 and high CD63 expression in both these databases, the Cancer Genome Atlas and the Chinese Glioma Genome Atlas, shows that high CD63 expression and high CD86 expression is actually poor prognosis. Those individuals actually have less survival time compared to those with low CD86 expression and low CD63 expression. And what our data suggests is that we can actually identify this in the blood itself. So whoever has high CD86 expression in the blood is likely to have high CD86 expression in the tumor, and high CD86 expression in the tumor is known to be bad. So we can just look at the blood and say these patients may have worse outcome. So let's treat them differently or more aggressively. That's what we are suggesting. Right, a quick summary. This is all in collaboration with Professor Namita Shah from Mazumdar Shah Center for Translational Research. This research also was done by this excellent student Jayashree, where we characterized myeloid cells in brain tumors. We identified highly heterogeneous neutrophil populations, and we suggest some blood-based biomarkers that can be used for prognosis. And this data is in this paper that was published recently in Oncoimmunology, and you can see it here. This is the DOI for it, right? So with that, I will end my talk with these last 2-3 slides that tell you a little bit about my lab. So the core expertise of my laboratory, as you've seen from this data, is core expertise in immunology, specifically in animal and human sample handling using flow cytometry and doing ex vivo cellular assays. We also have a materials group in the lab which works on scaffold fabrication, polymeric drug delivery systems, among other things. We're also now working on inorganic drug delivery systems. And we combine these two in preclinical animal model systems to actually test some of our hypotheses and see if we can develop better treatment strategies. If you think about it in a different way, so we also have clinical collaborators that are very seriously interested in doing clinical studies that involve both data and sample collection across hospitals in Bangalore. Right? But if you think of our lab pipeline, I like to look at it. We have projects in this immunology material sort of background, which some involve particles, some involve foot ulcers, some involve understanding neutrophil kinetics. We're doing mostly in vitro and in vivo work, but we hope to move into clinical studies and eventually develop product and technology that can be applied in the clinic, or at least applied in the market. We also recently have this work in oncoimmunology that is basically in the area of tumor immunology, which we hope to expand. It's primarily a clinical study. Now we hope to bring it back to in vivo models and see if we can develop better treatment strategies. And with that, I'll acknowledge all the different people who are involved. This is my group. All of this work was done by Preeti, Jayashree, Alkesh, Thiruvikraman, many others who are involved in this work, and they're also doing other pieces of work. This would not have been possible without our clinical collaborators. These were the clinical collaborators in all our diabetic foot ulcer projects. Wouldn't have been possible without collaborators within IISc, collaborators in MSCTR, Professor Namita Shah and her team, and all the funding sources. I'm primarily funded by the Intermediate Fellowship of DBT Welcome India Alliance now, but we've also had other funding sources in the past, right? With that, I will stop with this and leave you with this slide about BSSE. This is all about my lab, about the work in my laboratory, but some of you may be interested in what we do in the department. Our department is the Center for Biosystem Science and Engineering. We're basically the bioengineering department at the Indian Institute of Science. This is our website, which is getting upgraded now, so you can start to see what's happening. These are many of the students that are there, some of whom have graduated, and we're looking to recruit newer students. We, of course, have a major in bioengineering that has been going on since 2000. We're also starting an MTech program in bioengineering in August 2022. So if any of you are interested, please do apply to us. Research teams in our department are these, and this is continuing to grow. We are recruiting new faculty, and there are a lot of other things that we are working on. And so I'll leave you with this slide. I'll stop talking. Take questions now. Thank you.
Thank you so much, sir, for your valuable guidance. Participants, if you have any questions, kindly unmute yourself and you can ask your queries.
Don't mind, I'll stop sharing so I can actually see you all.
Yes, sir.
Yes. I see there are questions in the chat box. Should I answer them first?
No, sir. We don't have any question in chat
Oh,
box right now.
okay.
Yeah.
Okay. Thank you.
Delegates, I repeat, if you have any queries, you can unmute yourself and you can ask your question directly.
Okay. I guess if there are no questions, I'm happy to get questions over email too. So my email address is on my website. I hope you enjoyed this talk. I'm happy. I'll be here for another 15 minutes. If you have anything to ask me, feel free to. It's been an absolute pleasure from my side talking to all of you. I hope you enjoyed it.
Yes, sir. We enjoyed it a lot. I would like to take this opportunity to express my sincere thanks to our speaker, Siddharth sir. We truly appreciate your dedication for sharing your valuable knowledge with us. Thank you, sir. And we would like to take this opportunity to felicitate our guest speaker with an appreciation certificate. May I request, please do the honor, please, sir.
Thank you so much, sir. Thanks a lot.
Thank you, Dr. John.
Yeah.
This has been a pleasure. Thank you very much for inviting me.
Yeah, Professor Vandana is also with us.
Okay. Pleasure to meet you.
It was really a pleasure to listen to a very, very good piece of work.
Thank you.
I was thinking, Siddharth, is it possible, since now you have a link between glioblastoma and marker, can that be used for early diagnosis? Is there a kit available already?
No, that's something that we are actually working with this Mazumdar Shaw Center for Translational Research to develop. The first thing is that we are actually starting a fresh study to verify this. So this is a early study, right, which only 20 patients. So we need to validate this study when at least when we talked to BIRAC, they said, let's start with a validation study that's a larger cohort with at least 50, 60 individuals, according to the statistical testing. And then simultaneously, we're developing a kit for CD86 expression detection.
Yeah, it would be really great to see early on if we can know what are the chances of glioblastoma. Great work. Enjoyed listening to you.
Thank you for the wonderful question, and I hope we will be able to do that in the next 2-3 years.
All the best. We do hope that you will do it soon.
Yeah, thank you.
Thank you so much, sir. Thank you so much, ma'am.
That mail. Okay, so we have three.
Sorry, is there another question?
I think no, sir.
I think there are no questions from delegates.
Okay. Sorry, I finished a little early then.
Either they must have understood everything, or they're not, in a kind of research that at your level.
No,
There can be only.
Thank you. I mean, it's just I sped through some of those things. I was thinking there'll be less time, but thank you. This was wonderful to be able to speak.
Yeah. Thanks a lot, sir.
Thank you.
Yeah. Thank you.
Okay.
Thank you,